So, well, and, the other thing I didn't mention about REM sleep, but is actually clinically useful and used, is there are typically spontaneous erections during REM sleep. So I should just write that down. You know. We've talked about all these ways they are kind of compare and contrast. It's just probably something in the system to kind of keep things going, like kind of turn, basic maintenance or something. But it's used clinically, like if a male presents with erectile dysfunction and they're trying to figure out because it can be more psychological or it could be more physiological, and they want to know, like, is the basic physiology working to have the erection? What they do is they put a little strip of paper around the base of the penis at the beginning of the night. And if the guy has the physiology working, the little strip of paper will be broken in the morning, because when the penis swells with blood, it will break the little strip of paper. So this is actually used clinically as part of the assessment to see if kind of basic erectile function is working on a physiological level. So let's continue with kind of these big picture ideas about what's going on in the brain. Let's talk a little bit about like kind of memory and learning. So first I'll talk about two different types of learning that happen that seem to actually happen in different pathways, because you can actually have brain damage that affects one kind of learning but not the other. So there's the idea of declarative. Let me get a smaller. Declarative, also known as explicit. Why is this? Versus procedural. So known as implicit. So the basic idea here is declarative knowledge is stuff that you can declare. You can say, you explicitly know, like, I know that the receptor binds the ligand and then activates the G protein, which then travels through the membrane and it activates adenylyl cyclase. I can say it, declare. I have this explicit knowledge. I know that I know it. I can communicate it to you, what I know. Procedural or implicit is stuff that you learn, but you actually don't actually quite know that you know or that you have learned. Examples of like implicit learning, like they do these studies where they'll show you, there'll be a training session and they'll say, like, okay, here is a picture and this picture is a yes, and here, this picture here, that's also a yes, and this picture here, that's a no, and this picture here, that's a yes, and this picture here, it's like, that's a no. And you go through this training, and by the end of the training, you have no idea what is a yes and what is a no. It all just looks like, I don't understand what what they're getting at here, why one is a yes and one is a no. But then they'll do the next part and they'll just show you, all right, here's a picture. Is that a yes or a no? And you're like, I don't know. And it's like, guess. All right, I'll guess. It's a yes. All right, and here's the next one. Is that a yes or no? I don't know. I'll guess. It's a no. And you feel like you're just being random. You really don't feel like there's anything behind it, but you are actually there. It turns out there is some rule. There's some rule. It might be the yes has more density on one side versus the other side of the canvas or something. And you actually pick up on that, and you will start, your guess will actually be above chance. You'll actually be better than chance at knowing which ones have this property or not, but you don't know it. And you feel like you're guessing randomly, and yet you're actually choosing the things more often than not that is the one that actually does have the property. So that's this implicit learning, where you actually are noticing patterns, you're learning something, it's influencing your choices, but it's somewhere just in your neural networks. You don't actually have this explicit knowledge of what you know, or you couldn't tell anybody, why is this yes, or why is this one no? So that's this idea of declarative or explicit versus procedural or implicit learning. Like one of the kind of real, I think, clear examples of how these are different is one of the stories, and if people haven't read Oliver Sacks's books, he was a neurologist. He died a few years ago. He's amazing author, wrote amazing books. Almost anything he's written is amazing. One of his books that was one of his early ones was The Man Who Mistook His Wife for a Hat. And he talks about a patient who, in fact, we're going to talk about the hippocampus in a few moments, who has hippocampal damage and can't form new declarative memories. So whenever this guy gets some new information, or here's somebody, oh, hi, my name is David. And it's like, an hour later, it's like, oh, have I ever seen you before? It's like, so he has no ability to do this, but he still had this ability to form these implicit or this procedural implicit learning. Like they did a study where they, every morning, because he couldn't live on his own, he lived in a hospital, and every morning they would give him a choice, like, oh, you have to do some testing. You can go with Dr. A or Dr. B. And in the beginning, he would just pick, I don't know, Dr. A, Dr. B. He'd pick one and go with them. But Dr. A would always be mean to him, and Dr. A would always be mean. Dr. B would always be nice. But every time he met them, every morning he met them, they were two brand new people to him, because he could not form these kind of memories. And over time, he started always like, I can go with A or B. I don't know, let's go with B. And it wasn't because he had any memory that Dr. B is nicer, and but somewhere in his brain, he was still doing this implicit learning, and he was choosing, so when I think of things like intuition or something, this is how I tend to think it must be happening. Like, you are noticing things, you're picking up on things, it's actually influencing your choices, and you don't even know how you know what you know, but you just got a feeling, or you're going this direction instead of that direction, and it feels kind of random, but it's not totally random, because there is this kind of learning that is going on as well. So it's kind of wild. And we should talk about the formation, particularly of these kind of memories. Like, again, it's clear from some of these lesion studies, like with this patient I was talking about, that there seems to be different neural pathways involved between these two, because you can abolish this declarative but still leave this procedural intact to some degree. So let's talk now more about kind of forming memories and remembering things and putting things back, because that's been in more recent research a really fascinating area. So we'll So it's pretty clear that there are two main kind of ways things are stored in your brain. This short-term memory is when you're actually kind of holding it in the moment. You're thinking about it. You're kind of processing things. You're bringing things in, analyzing. Short-term memory seems to be more about kind of reverberating electrical circuits and stuff in your brain. This doesn't last that long. Long-term memory, there's actually physical changes, synaptic weights that get changed. And there's actually, like, they've shown, like, you can block long-term memory by giving drugs that block protein synthesis. So the neurons can't do whatever they need to do to make these physical changes in the networks that embed the memories in this more permanent way. Short-term memory, if you give somebody, ECT, electroconvulsive therapy, that's, they used to call that like shock treatment or whatever. It's still used clinically for like intractable depression. It can actually, I don't think anybody knows why it works. It's kind of like your computer's not working and you turn it off and on and it seems to be doing better. ECT, you put an electric current across the brain for a while and it somehow seems to clear out the depressive fog for people in a way that other things can't. But if you do this to somebody, you'll wipe out their short-term memory. Anything that happened in the last 20 minutes will be just gone. They'll have no memory. It'll just be evaporated. Anything that happened like an hour ago or longer, it's fine. So this idea that putting an electric current across the brain will wipe this out, but it won't affect this. That gives you another sense that this short-term memory is more about these kind of electrical reverberations going on, as opposed to long-term memory, which is something more physical. Again, kind of rewiring of neural circuits and things like that that are not going to be messed up by some random voltage going across your brain. Then what's interesting is, obviously, when you first are becoming aware of things, it's in short-term memory. It's in, it's coming into your consciousness as this short-term memory, but then you need to put it into long-term memory. This process, they gave it a name, it's called consolidation. Consolidation is the process of taking a short-term memory and creating a more permanent version of it. You can block this. Can be blocked like with drugs that inhibit protein synthesis. So that gives you a sense that there's actually some physical changes that are happening to create a long-term memory. Oh shoot, hold on. My iPad just lost its juice. Oh, please come back. All right, this is not good. All right. All right, I'm using my whiteboard that I'm running with my mouse right now, so this is going to be not as pretty. Okay, I had short term versus long term. And I talked about this idea of consolidation. Oh. And my, oh. Actually, it's going to be worth, I apologize for the delay. It's going to be worth me getting my iPad back online because it's just going to be so much more efficient. Yeah, what do you call it? We paused for technical difficulties. The nice thing is I'll edit it out of the video on YouTube so no one will ever have to know in the future. I just think it was a perfect day. All right. Okay. All right, so now we're back. All right, so short term, long term, consolidation. Oh my God. And now my start, oh, there, okay, now we're totally back. Okay, consolidation. This consolidation requires the hippocampus. So the hippocampus does lots of things. I'm not going to get into it. The hippocampus is a very highly studied part of the brain. But one of the things that it does is it is necessary to consolidate short-term into long-term memory. That patient that I was telling you about from that Oliver Sacks book, he had bilateral hippocampal damage that occurred from chronic alcoholism. And what happened was he could no longer form new memories. It's like that dude in Memento, if you ever saw that Christopher Nolan movie. Basically, he would meet you, he'd be charming or whatever, and like an hour later, if he met you again, it would be as the first time he ever met you. His whole self-image was still back from when he was like in his 20s. Like, you know, he had an autobiographical history, but it was up to a certain. Oh, can people hear me?
Yes.
Yes.
Yeah, I can hear you.
Okay. So hippocampus is essential for consolidation. Then what has happened in memory research that has been particularly fascinating is realizing that when you are going to then take something out of long-term memory and use it, so you have some memory that you actually want to use, that it's not like a filing cabinet that you just go in and pull out the file and look at it and put the file back, or, you know, peek inside and read it off. It's, you know, it's way or even like in DNA, right? If you want to transcribe a protein, you make an RNA copy of the master blueprint, which stays safely in the nucleus and pull the copy out into the cytoplasm. Instead, what happens is when you remember things, it goes from long-term back into short-term memory. So this is kind of remember. But then what happens is it gets reconsolidated. It gets put back into long-term memory. So whatever you remembered, and sometimes that memory gets like adjusted, like new information gets added to it or this or that. And this reconsolidation is this really trippy idea that your memories are actually constantly kind of mutating or evolving. Like there was a fascinating study where they had people talk about their memories of being small children at Disneyland. And the researchers, on purpose would try to contaminate their memories with, suggestions of, and eventually people would have these very firm childhood memories of being at Disneyland and taking their picture with the guy in the Bugs Bunny suit and all that. And, they knew that that couldn't have been a real memory because, Bugs Bunny's Warner Brothers. He couldn't have been at Disneyland. So they could have, they knew that these memories were not actually real, but what had happened was they took memories that they had as kids, and then the researchers kind of on purpose kind of messed with them a bit. And when they got reconsolidated, they weren't the same memories anymore. It's kind of a strange thing. Like you've probably noticed it yourself sometimes. Like you go back to some picture, it's like, wait, is that exactly what it looked like? Or you read something, or you're sharing stories with friends and you're not all agreeing exactly what happened. If you've ever been in a trial on a jury, it's just crazy how different people's memories of the exact same intense moment can be. But so long-term memory can be like changed by this. I should also mention hippocampus. We talked about being part of limbic system. So obviously emotional content actually makes a difference on how strongly things are moved from short-term into long-term. Most short-term memory just evaporates. They think part of sleeping even is, kind of separating the wheat from the chaff. Like there's so much stuff coming in, and most of it you don't need to remember. It just kind of goes away. One of the fascinating things that comes out of this idea of reconsolidation, as well as the idea that I mentioned that consolidation can be blocked by drugs that inhibit protein synthesis, is, at least on test animals, they have demonstrated that you can erase people's memory, or you can erase memories for in a mammal at least. Like they'll take, we have, there's two groups of rats. And both rats learn maze 1 and maze 2. So both mice, or both groups of rats have been trained on these mazes. Both are really good at them. And then what they do is they have them run the mazes. But here for these rats, they'll have them run this maze here, but give them, give the drug that blocks the protein synthesis. So I'm going to say that blocks reconsolidation. As they're running the maze. So they have to remember the maze in order to actually do it. So they're bringing up all the memories of how to do this maze and what do I do, left or right, where's the cheese or whatever. But as they're doing it, you've got this thing going. They're remembering the maze, but we're blocking the reconsolidation, so they can't put it back into long-term memory. So after this has happened, they lose the ability to do maze 1. And they still remember maze 2 just fine. You put them in maze 2, they can do it fine. You put them in maze 1 and they're like, oh, I don't remember how to do this. And the other rats that were just running the mazes but never got exposed to that drug that prevents reconsolidation, they still remember the maze just fine. So, it's an interesting idea, this idea of having, this kind of theoretical basis now to kind of try to go in and erase memories, or at least try to devalue memories. So it's something people look at. I mean, in some ways, some people are doing it in just with beta blocking drugs and things like that, things that kind of just keep your sympathetic activation down and try to have people remember something traumatic, but then have their body pharmacologically kind of modified so they can't get all freaked out by an old memory. And so then when it gets reconsolidated, the memory doesn't have quite that same edge that it used to be. So people are playing around with it in real ways, just kind of more mellow, like with beta blockers. But then there's also the more intense ideas about you could theoretically even erase memories, although what would that mean in terms of, we are kind of in some ways, who we are is kind of our autobiographical memory. So who knows? It seems like it could be good or it could be horrible. And it could be dangerous if you accidentally erase somebody's memory that's critical for who they are or something. La la la. So memory. This is still something we're looking at. I should also mention memory seems to be distributed. Like if you are looking, well, where is the memory located? It's not in one particular place. You can take a person and they can have damage to their occipital lobe. And the visual part of some memory will disappear. They'll still remember, being there at a certain time with certain people, but there'll be no component of a visual part to that memory. So it seems what memories do in some ways is kind of just reactivate the brain in some way, that the way it was activated when you were experiencing it, but in a less dramatic way. So you can tell, like, I'm actually just remembering it rather than reliving it, which is one of the issues with like PTSD, where people can't tell the difference between remembering and reliving things. So, but this idea that memories are not in one particular spot necessarily, they're distributed. I mean, it's a major, there's lots of active areas of research, but that's still something people are really looking at and learning new things all the time. Okie dokie. So let me think if there's anything else I want to say about, before we leave the CNS, I should talk about the spinal cord, because CNS is the brain and the spinal cord. So I'd feel kind of guilty if I did not at least say spinal cord is also part of the CNS. And again, some of the really important reflexes are integrated in the spinal cord, right? We talked about different spinal reflexes. Like the stretch reflexes. You don't need the brain. The brain can modify things, but the spinal cord has all the circuitry you need to kick out your knee when you smack on your patellar tendon. It has all the circuitry you need to pull your hand away from a painful stimulus. Spinal reflexes include ones we're going to talk about, like where you need to pee as your bladder gets full, or having an erection during like stimulation of the penis. You can actually have these fascinating, people who have spinal cord um, damage, who will be completely numb from the waist down, and yet they'll have an erection from physical stimulation of the penis because that's a spinal reflex. So the things are going here, coming out of here, even though nothing's coming up to the brain saying I'm feeling anything. So spinal reflexes don't need the brain. Gait reflexes, things of walking, like they've always kind of bummed me out, but these are these things where you take a cat and you put it on a treadmill, and then you basically like destroy its brain, and it'll still walk just fine as a treadmill goes. So basic gait, walking along, is something that can happen with circuits just in spinal reflex. So just giving the spinal cord some props that it is actually doing a lot of important stuff. It's obviously also a cable taking messages in and out with axons going, sensory in and motor coming out, but it also does integration as well. And then there's, the nerves that are coming off, cranial nerves, spinal nerves. I don't want to go, this is more an anatomy. What I will say, just as a just really brief superficial addressings of spinal nerves. The spinal nerves come off of different regions of the spinal cord. Clinically, why this is really important. We're going to talk about it from the autonomic level in a few moments. But when people get spinal cord damage, it's actually, especially like college age males, is like the most common way to get spinal cord damage, is like, do something idiotic and like dive into an empty swimming pool or something. So it happens, and depending where you cut the spinal cord, it is going to have very different effects on the body because of the spinal nerves that are then going up and down to the different parts of the body. So basically, at the very top, what we call the cervical plexus, this is where we have the phrenic nerve, and you should know the phrenic nerve. So coming off of here, they say C3 through C5. These are cranial nerves 3 through 5. These nerves are coming off of the spinal cord, and then they're making this phrenic nerve, and this phrenic nerve is what innervates the diaphragm. So when we get to the respiratory system, we're going to talk about the diaphragm being the main driver of pulmonary ventilation. I talked about, the pons and the medulla are important for these respiratory centers, but the message has to get to the diaphragm in order to drive the bellows in your chest, so to speak. So if you have spinal cord damage above this, you're pretty much dead, because you're going to stop breathing unless you're on a ventilator instantly. Like Christopher Reeve, when he had his riding accident, they were able to get him on a ventilator, but he was on that like for the rest of his life. Normally, they say the little mnemonic I've told people in anatomy, they say C3 through 5 keep you alive. So like if someone has spinal cord damage above C3, you're in a lot of trouble because you can't breathe. And it's different. Like when we get to the heart has its own little like circuits in there that keep it going. Like even if you rip somebody's heart out of their chest, their heart will keep beating in your hand for a little while. But you know, the lungs and the diaphragm, they need constant input from the brain down the phrenic nerve to keep going. Once you get below the cervical plexus, there's the brachial plexus. The brachial plexus goes down like to around T1. So it's still pretty high. These are the ones that go down to your arms. So if you like cut your spinal cord at the top of the thoracic region, you'd still have your arms. You know, you'd be a paraplegic. Your arms would still work, but everything below that would not be working. You know, obviously the lower you go, the more you, what am I take that back. So if you are above, so if you're like above the brachial plexus, you're a quadriplegic. If you're below the brachial plexus, you're a paraplegic. So just kind of, it's worth thinking about where these different parts of the bodies get connected to. Again, anatomy is where we go into this in detail. But just for today, just kind of, it's worth thinking about spinal cord damage because it's pretty common. People break their back and the spinal cord gets transected or something. You know, if you're below T1, you'll probably still have your arms. If you're, you know, around C5, your arms, nothing below your neck's going to work, but at least you'll keep breathing, you know, things like that. We're going to see that you end up having different things going on with the autonomic system, because the autonomic system is pretty unsymmetrical in how it emanates from the CNS. So we're going to see that in just a moment. So I'm going to just leave this. I just figure it's worth mentioning spinal cord because it's important. All right. We're going to now shift to the PNS. And there's obviously, what are the two main divisions of the PNS?
Somatic and autonomic.
Sympathetic, parasympathetic.
So you guys are all giving me subparts of parts, but I still haven't got the two main parts.
Sensory,
Motor and sensory.
vision.
Yeah, so the first thing to remember is the motor versus sensory. That's the first big division. And then motor was like somatic versus autonomic. And autonomic was sympathetic versus parasympathetic. So these are where we're going to head into now. We're going to start with the motor because it's going to be quicker. It's 9 o'clock. You know, we're going to be able to finish, I think, the motor to, you know, right now. Sensory gets more complicated because each of the senses, special senses in particular, is got a lot going on. But let's start with looking at the motor. And let's start by looking at the somatic division of the motor. And then we'll shift to the autonomic. So somatic motor. All right, we have the CNS. Where do the messages begin that are going to go out to operate your voluntary muscles?
Spinal cord.
Primary motor
Precentral gyrus.
Precentral gyrus. So we've got some like neurons in the precentral gyrus here. They're going to be sending messages out, and there's going to be some skeletal muscle here. Skeletal muscle. This is your voluntary muscle. So the things to know about the somatic motor wiring. One thing is there is always one motor neuron. The motor neuron has its cell body in the CNS and then travels all the way to the target muscle. So and it doesn't, I mean, and maybe I should said that a little weird. You know, you can have it could be in the spinal cord. It's just whatever neuron is going to eventually get to the muscle has its cell body somewhere in the CNS. There could be maybe more complicated wiring in the CNS, but ultimately, whatever axon is heading out along the nerve to go to the muscle, its cell body is in the CNS. It's probably in the spinal cord someplace. So one neuron goes all the way from the CNS. So one neuron goes all the way from the CNS to the target muscle, which is going to be different when we get to the autonomic, where you have a chain of 2 neurons before you get to the target muscle. Other things that are unique about somatic motor. The somatic motor neuron always releases acetylcholine. And then the target muscle, the skeletal muscle here, is always going to have the nicotinic cholinergic receptors. So what does that tell us about that synapse, that neuromuscular junction, if we know that it has cholinergic receptors?
It's always excitatory.
It's always excitatory. So when you fire off an action potential down a somatic motor neuron, it's always going to make the muscle twitch, contract. That's going to be different. When we get to the autonomic system, we're still going to see acetylcholine in some of the pathways, but sometimes it might make the muscle contract, sometimes it might make it relax. But if we're on the somatic motor, whenever you excite a somatic motor neuron to excite a muscle, it's going to make that muscle contract. And it has to do with the fact that we have these directly gated channels here. So any questions about that? Double checking something here. Yeah, so that nicotinic receptor can be blocked by curare. You've probably heard of curare. It's like what they used on like poison darts and stuff. This is an antagonist at this. It blocks it. So what would be the effect of giving somebody curare?
Muscle paralysis or
Exactly,
yeah.
muscle paralysis. The thing that you need to remember, and so curare is used clinically, like they use it sometimes if they're going to intubate someone. You want to shove a big pipe down somebody's trachea and you don't want them, we saw the gag reflex, you want to stop all of that. You want to make sure there's no muscle. What else do you need to do if you're going to give somebody curare to paralyze things before you intubate?
Do you need to affect their consciousness?
You need to do some kind of anesthesia as well, right? And there are some horrible, horrible stories of people who are paralyzed, but actually are feeling everything and excruciating, and they can't say anything because they can't talk or move or anything. And it's just, they're paralyzed and being worked on, and somebody may, spazzed and paralyzed them, but didn't actually anesthetize them in terms of the, not feeling things as well. So curare is also really dangerous because it'll paralyze your diaphragm. You'll stop breathing. So if
Professor Curare, it's a blocks the nicotinic receptors.
I'm sorry, I couldn't hear you. Say it again.
It blocks the nicotinic receptors.
It blocked, yeah, the nicotinic receptors. Yes. So
how do they keep the patient breathing though if they give them curare when they're intubating?
I think I'm not exactly sure how all that works. I think part of that
With a ventilator. It's a ventilator.
A ventilator. Okay.
Mm-hmm. I had it. That's why I know.
So yeah, no, we had this in the lab, and it always scared me because I knew like if you accidentally ingested it, you would just lie there, couldn't move, and wouldn't breathe. It was always kind of creepy to think about. So I think we are gonna be spending a lot more time talking about skeletal muscle and what exactly is happening. We are gonna come back here and revisit this neuromuscular junction, and then everything downstream from that leads to contraction, but not today. Other drugs though that are actually really common actually in Marin is like Botox. Botox works by inhibiting the release of acetylcholine from the motor neuron. So Botox stops this. Again, overall, if it was everywhere in your body, it's bad. You're going to stop breathing. You're going to die. But if you just inject a little bit around your eyes, then the muscles that are kind of tensing things up around your eyes are paralyzed, and you'll have less wrinkles, but you'll also lose the ability to have fine expression. You'll just look kind of like, you'll always look kind of like, I don't know. It doesn't make sense to me, but a lot of people do it. You'll lose the motor control. You'll lose the ability to have a little twinkle in your eye at the, no wrinkles, but no twinkles, I guess. Anyway, Botox works by inhibiting the release of acetylcholine at the somatic motor neuron. That's why it paralyzes muscles. again, which is different than curare, which blocks the ability of the acetylcholine to actually bind at the receptor. Right, so you can get the same end, why is this person paralyzed? There's different ways to paralyze people. All right. So now we're going to go to autonomic motor. So autonomic means like self-governing. These are the things that in general you are not consciously thinking about. That doesn't mean that you can't have any conscious control over autonomic functions. There's a lot of biofeedback techniques where people do get some level of control over autonomic functioning and all that. But in general, you're not thinking about, I better dilate my pupils, or I better speed up my heart, or I better like increase blood flow into this region of my body because it needs a little more oxygen, or better it's like it just happens. It's being controlled by your nervous system. There are a lot of reflexes, but not things that you are thinking about. So autonomic, there, like we said, so first I'm going to start with the things that are similar between both sympathetic and parasympathetic. And then we'll spend a lot of time looking at how sympathetic and parasympathetic kind of are different. So autonomic. What does it innervate? What is actually, we talked about somatic as innervating skeletal muscle, your voluntary muscle. What gets innervated by the autonomic motor?
Smooth muscle and cardiac muscle.
So there's smooth muscle, cardiac muscle, the involuntary muscles. What else?
Glands.
Glands. So this is turning on things like, and turning on and off, I should say. It's controlling. It might make you salivate more, or it might dry up your saliva. It might speed up your heart, or it might slow down your heart. It might relax your bronchial muscles and make it easier to breathe, or constrict your bronchial muscles and make it harder to breathe. So we're going to see it's not just excitatory like we saw with somatic. You can have a neuromuscular junction with the autonomic system where you actually relax the target muscle rather than excite it. So in both cases, you have two neurons from the CNS to the target. So let's say, let's just make the heart be the target. So here's your heart. And you're going to have one neuron. Let's make it black so it makes it easy to see. Here's some neuron. It goes a little while. Then it stops. Then it meets another neuron. Then another neuron continues to the target. What do we call cell bodies outside of the CNS?
Ganglia.
Ganglia.
So here we're going to have a ganglion of some sort. So we often call this one here the pre-ganglionic neuron. So oh, come on, silly autocorrect. So in both cases, sympathetic and parasympathetic, there's going to be two neurons, a pre-ganglionic neuron, a post-ganglionic neuron. But there's going to be differences in how this is all connected up and what kind of neurotransmitters are involved and this and that. So that is what we're going to talk about now.
I have a quick question. On the pre-ganglionic neuron, it's just starting in the spinal cord. Does it start up like higher in the brain, or?
So I'm going to talk about that specifically in just a moment. It depends if you're in the PNS, in the sympathetic or parasympathetic.
Awesome, thanks.
Yeah. I should also, let me just mention this this synapse in the ganglion. This is just kind of a relay. So this just kind of keeps the message going. And this is just a cholinergic synapse with nicotinic receptors. So what that means is cholinergic synapse, this neuron here, this pre-ganglionic neuron releases acetylcholine. That acetylcholine is recognized by the nicotinic cholinergic receptors, which are always excitatory. So you send a message down here, it's continuing on here. So it's more of just like a relay to keep the message going here. When we get to the target, we'll see it's much more complicated. But the synapse in the ganglion, you can just think about just like keep the message going. Right. So now what we're going to do is we are going to look at the differences between the sympathetic versus parasympathetic. I'll try to keep it kind of color coded as well. Sympathetic, that's the fight or flight. That's like speed up your heart, dilate your bronchial passages, bring your energy resources online, get sugar into your bloodstream, like get ready for danger. That's why I kind of made it red. Parasympathetic, again, people talk about the rest and digest. Let's get digestion going. Let's start salivating. Let's encourage digestive processes. Let's slow down the heart. Let's constrict the pupils. For the most part, these tend to be antagonistic in terms of their activity. So I'll say mostly antagonistic. Meaning sympathetic speeds up your heart, parasympathetic slows down your heart. Sympathetic dilates your pupils, parasympathetic constricts your pupils. Sympathetic sympathetic will inhibit, shut down digestive processes. Parasympathetic will stimulate digestive processes. So, and it's also, like I think I mentioned a while ago, it's not like all or none. There's like driving with a foot on the brake and a foot on the gas, and you're constantly just kind of adjusting, depending just as you're moving more, you might need to speed up the heart to keep your blood pressure going, and then you're resting, and then more parasympathetic slows it down. So it's not like you're in one mode or the other mode. They're both working together. You walk into a bright room, parasympathetic is the one that is going to be in charge of constricting your pupil. That was weird.
So Dr. E, where does freeze come in terms of sympathetic or parasympathetic?
Where does what?
The freeze response with like fight, flight, or freeze too? In terms of like trauma responses too, of like the paravagal nerve, and I'm just
So
I-
the vagus nerve is typically parasympathetic. I would have to look more. I'm not exactly sure.
okay, yeah, I'd be curious.
Yeah. So mostly antagonistic. There are some exceptions. Exceptions, sweat glands. Just sympathetic. Let me spell that properly. So your sweat glands, if you have more sympathetic activation, you sweat more, less sympathetic activation, they turn off. So it's not like a push and pull. So this is actually used in a lot of physiological experiments to monitor sympathetic activity. The more sympathetic activation you have going on, the more your sweat glands turn on. And the more you sweat, like if I'm looking at somebody's hand, and I look at the, electrical resistance. What is the electrical resistance here? The more you sweat, salt water conducts electricity, the better your skin conducts. So they call it electrodermal conductance or galvanic skin response. So by looking at the resistance across your skin, it's actually a nice measure of your sympathetic activity. This is part of what they use in lie detectors. If this is where it's kind of sad not to be in lab. We actually have one of these little galvanic skin response things that we can use in our lab. People have used as part of their independent research projects. And you look at resistance, it's at something, and you go and I've done this. You just walk up to the person who's hooked up to them and then you kick them. They're like, what the heck did you just do? And all of a sudden it's like, you see this will be conductance. The conductance starts going up because as they start having sympathetic response to getting kicked, the sweat glands fire off. More sweat is more salt water, better conductance, and you just see this thing go up. And then eventually it kind of comes back down. And then they just keep sitting there and you ask them, so did you have sex last night or something? They go, what? And it goes up again, and then it starts coming down again. So it's fascinating. It's really it's easy to observe. If you just make them physically or emotionally kind of stressed out, you get this real clear spike in skin conductance, which is resulting from increase of sweat. So just putting that out there, that's used, it's probably more research than clinically, but it's fun. It's fun to mess with. And again, it's used in lots of spy movies or stuff because it's at a core part of the lie detector test. The polygraph, is like they look at different things like your heart rate and this and that. But part of the polygraph is they call galvanic skin response. GSR. So sweat glands, most blood vessels as well are also just sympathetic. We're going to talk more about this when we get to the cardiovascular, but your blood vessels have smooth muscle, and the more sympathetic, the sympathetic activity can will relax or constrict to adjust the diameter of the blood vessels. It's not, there are some things that are parasympathetic when we get into sexual response, like the vasodilation that results in erection is actually from parasympathetic activation. But I will say most blood vessels are purely sympathetic. When we're going to talk about the whole vasomotor system, when we look at, kind of neural control of blood pressure, that's mainly a sympathetic innervation that is constricting blood vessels to try to, keep your blood pressure up and stuff. The other thing, actually talking about sexual response, that's one of the places where these kind of work together rather than being antagonistic. Like in sexual response, parasympathetic is responsible for the vasodilation leading to erection. But then at some point at the height of kind of sexual stimulation, there's like orgasm. And that actually is this shift from parasympathetic to sympathetic activity. And this is what's causing the ejaculation and everything. So you kind of need both of them working together to have that go. So they're not fight. What can happen is the sympathetic can fight the parasympathetic, which was why getting nervous, like a guy can have trouble getting an erection if he's like kind of too nervous about things, because then that sympathetic is not allowing the parasympathetic to do its job. So let us now go back to, let me erase all this stuff. So here I've just been talking more kind of functional stuff. All right, we're almost done erasing all this. There we go. So now what we're going to do, let's see, all right. If we have the brain and spinal cord, I'm going to use the heart as a target, just as a kind of, target for the autonomic nervous system. It seems like a good one. Again, parasympathetic would slow down the heart, sympathetic would speed up the heart. So some of the ways these are very different is even where the neurons begin. Parasympathetic is what we call has a craniosacral outflow. Nabbit. Meaning that the nerves, the axons that form the parasympathetic system, most of them are arising from the cranial nerves, just straight off the brain, and then also just from the sacral region of the spinal cord. In fact, something around 70% of parasympathetic innervation is coming down. What's the only cranial nerve that wanders out of the head and goes into the body?
Vagus nerve.
Vagus nerve. So 70% of the parasympathetic innervation is coming down the vagus nerve. Cranial nerve 10, the vagus nerve. Vagus nerve does other things too. It has motor stuff, it has other things, but a big part of what it does is carry a lot of parasympathetic motor stuff. Sympathetic, on the other hand, we call the thoracolumbar outflow. Oh, I want it red. So in this case, you have the neurons or the axons arising from the thoracic and lumbar region of the spinal cord. This has implications for spinal cord injuries as well, right? If you have some spinal cord injury that cuts off, at the top of the thoracic region, you are going to be just fine for most of your sympathetic innervation, but you're going to be fine for most of your parasympathetic innervation, because the majority of it is coming from the cranial nerve, which is not being affected at all. But you are going to wreak havoc with your sympathetic. And depending where you cut it, supposedly, like if you have a spinal cord injury at a certain level along the thoracic, you'll just be able to sweat down to a certain level of your torso, and beyond that, the sweat glands don't work anymore. So having this, it is important to realize where these are coming from, because it makes a difference when you start thinking about what are going to be the kind of things that are going to happen with different spinal cord injuries. So sympathetic is coming from thoracic and lumbar regions of the spinal cord, parasympathetic, cranial and sacral nerves. And again, the majority coming from the vagus nerve, which means it's not even needing to come down the spinal cord. This one just kind of comes out the foramen magnum and starts zipping around. Oh man. All right, so we're going to continue looking at sympathetic. Whoops. I'll do my color coding, sympathetic in red. Sympathetic versus parasympathetic. Sympha. So far what I have said was sympathetic is the thoracolumbar outflow. Versus the parasympathetic, which was that craniosacral outflow. Another big difference is the length of the pre and post-ganglionic neurons. Parasympathetic, you have a very long pre-ganglionic neuron. And short post-ganglionic neuron. What that means is the ganglion is right near the target organ. Ganglion is near the target. Whereas in the thoracolumbar outflow, it's going to be a short preganglionic neuron, long postganglionic neuron, that means the ganglia are near the spine. So if you've been in anatomy, you remember the whole sympathetic chain ganglia going up and down near the spine. So if I draw it out, it's like this. Like here's the brain and the spinal cord. And like I was saying, we had like if the heart is supposed to be like our target organ here. In the so sympathetic is going to be starting in the thoracolumbar region. There'll be a ganglion right near the spinal cord there, near the spine, and then a really long one that goes all the way to the target. Whereas when we're talking about parasympathetic, craniosacral is starting as a cranial nerve, and then it's basically going almost all the way to where it's going. And the ganglion is basically right near the target. And in fact, sometimes the ganglia are even within the wall of the target organ. They call them intramural ganglia, or sometimes they call them terminal ganglia because terminal at the end of the line. So parasympathetic ganglia are near the target, sympathetic near the spine. And then the big thing is the post-ganglionic neuron. This synapse here is going to always be acetylcholine if we are in the parasympathetic. Oops, I want it blue. And the target is going to obviously have to have receptors for that, but it's going to be different than we saw in the somatic motor system. Target is going to have muscarinic cholinergic receptors. Well, not receptors. It auto corrects me when I don't want it to, but then when I do make a genuine typo, it doesn't. All right. So what does that mean? If we have post-ganglionic neuron releases acetylcholine, the target. So what is that? What are the implications if the target has muscarinic versus nicotinic cholinergic?
That has second messenger. It could be excitatory or inhibitory, depending.
Yeah, exactly. So that's important to remember. You have to go case by case. Like when you activate this motor neuron, is it going to contract or relax the target muscle? It depends on the case. It's going to stimulate. Parasympathetic will stimulate your digestive tract, but it will inhibit others, slow down your heart, right? You really need to look on a case by case because a muscarinic cholinergic receptor is mediated by a second messenger pathway, which has lots of flexibility, right? You can have lots of outcomes after you bind the receptor. Sympathetic, on the other hand, post-ganglionic neuron, typically, I'm going to say typically releases, we'll see there's some exceptions, releases either norepinephrine or epinephrine. The target, has a variety, has adrenergic receptors. And there's a variety of different kinds of adrenergic receptors I was talking about. There were the alpha adrenergic and beta adrenergic and alpha 1 and beta 1 and beta 2 and this and that. For this class, you do not need to know the details about the different kinds of adrenergic receptors and what they do. But again, like, beta, and we talk about beta blockers. Beta blockers are blocking beta adrenergic receptors on your heart. And so when this neuron releases its norepinephrine there or whatever, it's not going to speed up your heart, even though you're like, oh my God, I just saw a tiger. It's like your heart's just going boom, 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 because the message to like speed up isn't getting there. A beta blocker would be a beta adrenergic antagonist. Meanwhile, if you are at an eye exam and you want to be able to shine the light in your eye, but you don't want the pupil to constrict, right? For an eye exam, the eye doctor has to shine a light in your eye and look around in there, and he wants to see. He doesn't want your pupil going down to a pinpoint. So if you wanted to block the pupil constriction, what kind of drug would you have to use here?
Acetylcholine antagonist.
Exactly. A cholinergic antagonist, something that blocks the cholinergic receptors. So the message from your brain saying constrict never actually activates the muscles that operate your iris. So just kind of, so there's a whole bunch of different drugs. Again, this is going to be later on when you get into your pharmacology and stuff. There's a whole bunch of drugs that are specifically to bind, to either block or activate sympathetic or parasympathetic. And the different subclasses of receptors tend to be localized on different areas. So, you actually have some amount of finesse in how you are modulating the activity of the sympathetic and parasympathetic systems using the different drugs that are available. So, that's why it's important to really understand that at a fundamental level that they are using different neurotransmitters, and we have pharmacological tools that can go in and preferentially block or activate one or the other side of your autonomic nervous system. Right? If someone has an asthma attack, you want to dilate their bronchial passages, you give them albuterol, which is an adrenergic agonist, something that is going to activate the adrenergic receptors, get the sympathetic thing going. I say typically there are some exceptions that you don't need to worry about. Like sweat glands actually use acetylcholine, even though they're sympathetic. But for the most part, to a first approximation, you can think about sympathetic being epinephrine and norepinephrine. The other big difference between these two is sympathetic, in addition to operating along these motor pathways mediated by neurons, can also trigger the dump of adrenaline or epinephrine by the adrenal gland. So that's the other thing that this side can do. Can release epinephrine from the adrenal gland, from adrenal medulla. So this is really different than sending a directed message like from the CNS to the heart, it's time to speed up. When you have the sympathetic nervous system tell the adrenal medulla, just dump epinephrine, dump adrenaline into the bloodstream, this goes into the bloodstream. Now it's everywhere. And every cell that has the appropriate adrenergic receptor is going to have a response. So your eyes will dilate and your heart will speed up and your bronchial passages will dilate. All that's, so everything happens all at once. Again, that's the definition of a hormone versus kind of a neurotransmitter just being released at a synapse. We'll talk way more about that later in the semester. The parasympathetic doesn't have anything like that. But in some of these, again, for this class, we're not going into the detail. Some of these adrenergic receptors bind epinephrine and norepinephrine equally. So they'll either respond to the direct neural pathway or they'll respond to the hormone dumped into the blood. Some of them are optimized for one or the other. But in general, you can think of them doing the same kind of thing. And again, what's the other word for epinephrine?
Adrenaline.
Adrenaline.
And we'll talk about, like when we get to the cardiovascular system, we'll talk about EpiPens, like why you jam a little EpiPen into somebody's thigh if they've got some anaphylactic shock and their blood pressure's dropping to 0 or something. It's basically part of the sympathetic nervous system is going to bring your blood pressure back up and constrict your blood vessels and speed up your heart and bring your blood pressure up if you've gone into the systemic vasodilation. What else? Any questions about autonomic? I think I've covered it.
I have a question about like anticholinergics, like those kind of medications. So they would affect both sides, but have a greater effect on the parasympathetic sides, and they wouldn't be affected by like adrenaline or other hormones.
Anticholinergics are blocking this, so like they would like kind of dry up your secretions, like your salivary glands and stuff like that. And then some of them, like the datura, like Brugmansia. I mean, you've probably, do people know what like Tridatura, Brugmansia are?
No.
Sure. So they're in the Solanaceae family of plants. I'll bring a picture of them up because it's worth actually knowing what they look like. So you know, there are plants you can nibble on and they're food. There are plants you can nibble on, they're just not so great. Or some plants you nibble on and they're kind of nice, they give you a buzz. And there are plants that will really mess you up. And there are really common plants that are with these anticholinergic drugs in them that will really mess you up. So it's worth getting to know them. So these are all over the place. I'm sure you have seen these before. They're beautiful. I love them. And at night they smell, they're so intoxicating. I have a bunch of these growing around where I live. But you don't want to eat them because they will make you like delirious and then could kill you at higher doses. At the same time, they were used like even back in Roman times at low levels for here let me go to Belladonna. So Atropa belladonna. So belladonna, it means beautiful woman. And it was because they used the Belladonna Alkaloids that, which are anticholinergic, and they'd use them as little eye drops to dilate the pupils. And that was, considered beautiful. You'd walk into the room with these big, wide pupils. So it was used. And the problem with dilating your pupils is it totally messes up your focus and everything. Like, so you walk in and you're at the height of your current like womanly fashion, but you can't focus on anything. And you can't constrict in bright light and everything. But Belladonna alkaloids are something you should know about. They are in Datura and Brugmansia and a lot of these things. And like this is the family that has tomatoes in it as well. Solanaceae. The witch's herb. I think because again, at lower doses, it can make people not dead, but just really kind of delirious. But it's not something to play around with. Yeah, I mean, actually just the Datura. This Thorn Apple, you probably, Thorn Apple is the common name for, you often just see this thing just growing around. It's got these beautiful flowers, and then it has these crazy fruits. That's why it's called Thorn Apple. There it is. I mean, this is growing all over the place. But again, if you see it, don't eat it. But they smell great.